Welcome.
The Pathology Innovation Collaborative Community is a regulatory science initiative that aims to facilitate innovations in pathology, as well as advance safety and effectiveness evaluation. The PI Collaborative Community aims to harmonize approaches to speed delivery to patients through collaboration in the pre-competitive space. The PI-CC is open to all stakeholders, public or private, including but not limited to academia, industry, healthcare providers, patients, and advocacy groups. Meetings of this kind are an example of lawful activity under federal antitrust law, because the focus of the meeting is on government action or policy, including the industry's responses or positions taken with respect thereto. However, antitrust monitoring is needed for companies' own protection, since they are direct competitors meeting together. These meetings need to stay within protected subject matters and need to be monitored so that they do not stray off into inappropriate areas, such as pricing and price terms, sales and service territories for particular products, customers and customer territories, each company's individual decisions regarding selection of suppliers or customers, marketing plans and especially future marketing plans or new product offerings, other proprietary or competitively sensitive information. Today's meeting will follow the written agenda, and there will be no discussion of pricing or other prohibited topics. If the discussion veers into those topics, we will terminate the discussion. If you have any questions or concerns about the propriety of a discussion, please raise it immediately, and we will take a break to determine how best to proceed. The Medical Device Innovation Consortium acts as the formal convener, and we will follow their conflict of interest policy. For additional information, we defer to our Code of Conduct and our website, www.pathologyinnovationcc.org. We look forward to moving the field forward.
Thank you very much, Ulla. Okay, welcome to the first session, OB Scout. It's a mouthful of an acronym, and I leave that up to the two principal investigators, Ali and Hossein, who triggered this. Briefly, this falls into the big topic of grant challenges for potential use or secondary use of data for downstream regulatory purposes, and I'm very glad to see some of the expert faces here today. And without further ado, I hand it over maybe to you, Ali.
Yeah, thank you so much, Joe, and thanks for everybody who has joined so far. So we put together a few slides for background, and then I think then it will be probably like 5-6 minutes, not too much, so that we have more time for discussion. So, okay, so let's see. Where is the page? What I'm going to share. Okay, let's see if the presentation mode works. Do you see the presentation mode? Yeah, okay, that's all good. Okay, so what we have in mind, so this is the name, sort of like an acronym for the project that we have in mind. So obviously up for suggestions if you have better naming. So we're more than happy to consider that, but this is just preliminary anyways. So so the idea is to sort of like build on what Panda has done. So they've done a great job, but then we want to build on it and learn from those, the things that we can actually improve. And also, I think the key aspect that we wanted to include within this Grand AI Challenge is actually the regulatory aspects. But I think, that'll be absolutely important to take things actually to the next step. So, so the focus area is on ovarian cancer, and then just a quick background on ovarian cancer. It's the deadliest cancer of the female reproductive system, and clinical diagnosis is mainly done on microscopic examination of the H&E images. So epithelial ovarian cancers have 5 major histotypes, which include clear cell carcinomas, brain carcinomas, endometrioid, low-grade serous, high-grade serous, and mucinous carcinomas. So distribution is sort of like high-grade serous is around like 70% of cases represent high-grade serous, the most aggressive one, more or less. Then CCOC and endometrioid represent about 10%, and low-grade and mucinous roughly like up to 5% each. So this was actually a discovery that was led actually by a Vancouver team and Dr. Blake Jilts, who's in charge of the WHO guidelines for gynecological cancers. So so he's been actually working on this project with us. So why these subtypes matter, because they have different etiologies, molecular profiles, and clinical outcome, and treatment pathways are beginning actually to become more histotype specific. But the challenge is that inter-observer agreement in histotype classification is low to moderate. General anatomic pathologists, they get to cap a coefficient of somewhere between, like, depending on different studies, between 0.54 to 0.67. Trained gynae pathologists, subspecialized pathologists, they actually get quite good concordance. So that is 0.89. For that reason, there has been, other like orthogonal tests or ancillary tests like immunohistochemistry, and potentially people are exploring adding gene expression profiling so that they can actually increase this agreement, or in other words, make a diagnosis better. So what we have in mind is actually to work in this, to start a challenge in this area. So we have actually a couple of publications, and one is actually going to come out in Modern Pathology hopefully soon. It's been accepted, and we've done this study on about 500 patients, local patients, and also an external cohort. So here comes the challenge of OV Scout. And so what we have tried to do is we've gone through the Page AI's regulatory submission. That was the public part at least, and also the Panda challenge, which I actually wrote an editorial on it in Nature Reviews Urology as well. So anyway, so we've done those analysis, and then we've come up with sort of adapted some of the things that have been covered in Panda, as well as the Page AI's regulatory submission. So so the idea is obviously to make this in a way that we can eventually use this data or make it public, so that others can actually use it for regulatory submissions as well. So that's the end goal. If we can do it, that would be fantastic. So it's going to be an in vitro diagnostic medical device software. Let me see if I can minimize. There's one screen that shows up on my end. Okay, that's better. So, and it's going to be a potentially deep learning based package. So we'll see. I think most likely that's going to be the case, like the Panda challenge, that all the submissions were based on deep learning models. Input is going to be digital pathology, obviously slides, and then sort of like maybe a cloud-based system, or that can be cloud-based, either in a local cloud or commercial cloud providers as well. And then the output is going to be one of the 5 histotypes that I mentioned, and then that's going to be used to augment pathology. So meaning that it will provide a diagnosis for a pathologist that can review and sign out. But what we want to do is actually to take this one step further, is that not only we want to do the 5 major histotype diagnosis, we want to identify the outliers. What that means is that, in a real practice, so what was, I think, one of the key areas that we need to focus is that if a case is suspected to have, as an example, like ovarian cancer, or it can be applied to other cancers as well. So it's suspected of having ovarian cancer. If we want to take it to the clinic and make it more, I think, clinically applicable. So a lot of those slides or patients may just have benign benign tissue, or like normal fallopian tube, or other rare subtypes. So in other words, the 5 major histotypes that we are talking about, it's what if these cases are metastasis from other sites that we see in the ovary? So they shouldn't be classified as the 5 major histotypes. So having that in mind, so we want to set the challenge to have actually 2 specific goals or tasks. One is just imagine it's the input is going to be one of the 5 histotypes. We know it's one of the 5. That's where the challenging cases are. And then and then we want to diagnose the 5 major histotypes. So, but the second one is actually identification of outliers. And again, these are cases that are suspected to have ovarian cancer, but do not fall within the 5 histotypes. So that will be the second challenge or second task. And in this case, we're going to provide some outliers like metastasis from other sites to ovaries, and rare subtypes of ovarian cancer, which I will go through those tasks a bit in more details. Okay. So this is sort of adapted from the Panda challenge and also Page AI's regulatory submission. And so the numbers bit arbitrary at the moment, but we're going to do power analysis to just make sure that all the numbers make sense and we have enough statistical power. We have done that for other grants, so we know that these numbers are fine, but still we're going to do that to just make sure everything is fine. And so we're going to have a training data set and a tuning data set that, like the Panda challenge, so the developers within the development phase, they can build models based on the training data, and then the tuning set, they can get sort of like scores or performance sort of feedback that can be actually incorporated in their model training. So this is the only place that this is the phase that they will have access in other words to feedback. But then we have the validation phase that includes 3 sets. And this one is again adapted from Panda, but also from Page. We have included some other aspects that we feel like it's important. And again, up for discussion and feedback. So within the validation phase, obviously this is blinded to developers. So imagine some people are going to build models here, get feedback through tuning set. At some point, they're happy with it. And then, we're going to have a leaderboard. So we're going to select top performers, whatever that's going to be. So top N, we will come up with that, maybe 10, 20 teams. And then we will take those algorithms and then apply them on 3 different data sets. So this part is again adapted from Page. So what we're going to do is that we're going to have part of the tuning set, it will be this test set 1 is going to be the tuning set, but we're going to scan it with different scanner, or maybe a couple of different scanners. And that's sort of like a bit of more of like a stability test as well. Set 2 is going to be, let's say, roughly maybe 500 patients. That's going to be from external sites, potentially 3 sites. We can have more. And this is going to be a combination of whole sections and also tissue microarrays. Because for tissue microarrays, we have access to a lot of patients through the ATA consortium. So, and also we're going to have a 3rd set, which is roughly maybe 60 patients. That's we're going to do stability testing. This is what Page did, but they named it differently. But we're going to scan the slides with different scanners, potentially at multiple time points, to just again to account for variability in the scanning, operators, scanning time, and different scanners to see how the algorithms are working within that setting. So then for task 2, the only difference is that it's going to be exactly the same as task 1 in terms of development phase, validation phase. What we're going to do is that we're going to add potentially 25 outliers to the tuning set. And this is going to be, again, representing whole different things like, mets to ovary, rare subtypes of ovarian cancer, and also normal tissue as well. That's going to be the tuning phase. Within the test, validation phase, we are going to add outliers over here. And we may actually, obviously those 25 are going to be part of this one as well. We're going to have outliers here, 50 outliers here. For the stability testing, we may actually include, some outliers as well. So that's sort of as far as it goes for the challenge, but then the clinic, what we have in mind, this is what Page did. So they had a clinical validation as well, and the question was more about, what is the difference between like AI-assisted diagnosis and unassisted diagnosis. We want to see, if let's say we'll have to again come up with how many winning teams are going to go through this evaluation phase, because this is the part that we are going to show. We're going to have pathologists that are going to do unassisted diagnosis, let's say on 100 patients. Again, numbers can change based on power analysis. And then followed by AI-assisted diagnosis on the same cases. Because now at that point, we just show the diagnosis and see, if they change their diagnosis. And then obviously we have the gold standard, meaning that we have the labels that have been reviewed by multiple pathologists, and also like, immunohistochemistry has been done to confirm the histotype. So we have fairly, I think, good confidence that the labels are correct. So again, so number of pathologists, number of the sites or patients are going to be up for essentially for power analysis to just make sure that we do it right. But this is going to be also a blinded, obviously, validation that we have in mind. So that's essentially, yeah. So that's the design that we have in mind. And then now I think we can start the discussions, and I can stop sharing for the time being if that's easier. If need be, then I can share the slides again.
So maybe before we get to the questions, and Ali, I give you a minute to read, because there are a few in the chat.
Oh.
I just wanted to briefly point out that we're hosting a session to go over a decision summary, the one from Page that you mentioned. Ulla, it was June 15th, is that correct? No?
The 22nd.
22nd, yes. Announcement is coming where we will have someone from the Office of Policy from the FDA joining to explain the structure of a decision summary. It will not be, of course, focused for AI grant challenges, but to just illustrate some of the things that can be learned from the decision summary. I just wanted to point that out. So it's somewhat related, true, but unrelated. Some of the points that you mentioned, Ali, may be helpful just for the other folks on the call. Brandon, I see you have put in a few questions in the chat.
Okay, so maybe I can go over the questions and then one by one. So the first one is, sizing what will be the performance metric. So that's a good question. We should have included that, but I think we're going to go with kappa, but also balanced accuracy. So those are the two that we have in mind. But do you have any other input?
I would say not kappa.
Mm-hmm.
It is not a reproducible metric. Your sampling cases are, I don't know how close they're going to be to prospective sampling in the clinical population, but that's the sort of stuff that really messes with kappa. Is any study has a different distribution, and all of that is confounded in the kappa statistic. So I think I saw 5 or 6 types, tissue types. Is that right?
Yes.
Is it 6 or is it 5? I can't remember.
So it's 5 histotypes with the outliers. It's going to be 6.
Right. So basically, think about that 5x5 table. I mean, that's the raw data, right?
Yeah.
And you're going to have one of those for each algorithm. But so for one algorithm, you're going to have that algorithm compared to your truth. And then, you know, there is a sensitivity and specificity by category that you can do.
Oh,
And that, you know, sensitivity and specificity, is something that's reproducible, because, I mean, at least if you sample within those categories well enough. Whereas, and I know everybody likes to have one number as the final answer, but that just does not characterize algorithm performance, especially in this multi-category world. Now, I can't tell you what to do with your challenge, and I know you're going to be driven to a singular number in the end, because everybody always says that's the only way we know how to determine the winner. But I hate that argument. Sorry. Because the characteristics of the algorithm is the performance on each one of those types. And so maybe you do add them all up in the end, but if you want to figure out which is the most important and order that into a multiple hypothesis test that has a more statistical rigor to treating each one of those categories. That's my main suggestion, and that permeates everything, especially when you start talking about what is your truth.
Right. Yeah. I think that's a great point. So, at the end, yes, we're going to have the confusion matrix. It will boil down to how we rank them. But you're right, it could be like a 5 category. So the challenge obviously is that, and then we will talk to pathologists again to make sure that, you know, if we do a sort of like a balanced sort of like averaging, that would be actually something that is reasonable to do. And yeah, so especially that these are going to be like 70% is high grade serious, then we have 10, 10, 5, 5. So it's pretty imbalanced.
Right. And you might even think to make your sample set balanced. You know, they call that stratified sampling, where you get an equal number of each of those categories. That helps all of the metrics, and it helps size your study, because then those rare events won't seem rare in the study. They'll be just adequately sampled, and your performance on that category will have confidence intervals commensurate with the others.
Right. Okay. Yeah, that could be on I think we can do that on our test set.
That's it, right.
To just remove that variability in terms of imbalances.
I'm guessing on the training set, you're just gonna push out as much data as you have.
Yeah. Because otherwise, I mean, the rare subtypes, like, you know, like low-grade serous or mucinous carcinomas, you can't get like thousands. It's going to be impossible to get that. Yeah. It's going to be in the order of like probably hundreds. But for high-grade serous, it could be like, you know, 2,000 cases that we can put together for various parts.
So Brandon, I just wanted to ask again, so for each of the 5/6 subtypes, what would be the appropriate metric, so to speak? Agreement or?
Well, there would be 5 metrics.
Yeah, no, but each one of the 5 metrics.
If you have a singular truth or reference standard, let's call it reference standard, because the best use of that term here, you would have a sensitivity and a specificity for each one of those. But you would have a sensitivity for each one of them, and specificity is kind of captured by having that multi-parameter performance metric. So you don't really need to calculate specificity, because it kind of plays its role in all the other categories.
Mm-hmm. So it's basically 1 against the remainder. Got it. Mm-hmm.
Basically, you create a 2x2 table for each category.
Yep.
Okay, any other input for this question from others? If not, I can move on, to the next one. Okay, hearing none. So the second one is, okay, what will be your true labels? So these are, good question. These are going to be, these are the cases that have been reviewed by multiple pathologists, and for the cases that are sort of like, in question, additional tests have been done, which includes immunohistochemistry. So for that reason, we can treat that as sort of like, that variability. Otherwise, we see like intra-observer variability is going to be gone in these cases, because again, they're fully reviewed by multiple expert pathologists.
So I would just say to write up the protocol for determining truth. You say that the variability is going to be gone. I highly doubt that. Now I know in the patient record, the final decision is the final decision there, or a final record. But if you have multiple pathologists, they're not going to agree, right? So how you determine that result, how you aggregate those numbers, is it a majority rule? Ultimately, here at the FDA, we would ask for some characterization of the agreement of the people that are doing your reference standard. And I'm not saying all this has to be done for your challenge, but at least documenting really well so people get that clarity. And you can then have that as your part of your arsenal of defending what you did. But I think writing that down is always very essential to transparency.
Yeah, absolutely. I think that's a great point. Then we can write that up. So eventually it's consensus based, but as I said, because they're doing the additional tests, those are the parts that will sort of help with the diagnosis as well. If they just look at the H&E slides, yes, there's going to be disagreement. But when immunohistochemistry comes in, that will sort of like help a lot. And that's what people are actually doing in, like, let's say in hospitals that have access to immunohistochemistry, they can't tell, they just order IHC. Then that gives them like sort of like much better diagnosis. But it's an important point to document that process anyways. And whether the training is going to be including TMAs and whole sections. For training, we're not going to include TMAs. It's going to be just for the testing phase that we're going to include the TMAs. And because I think we have enough training data from whole sections and enough diversity that people can build the models off of those. And so the other challenge that we have is that, like, we have been talking to Kaggle to host this as well. And there is limitation in terms of the data storage. So that's also a factor. But for training, we are going to go with WSLs. Testing, TMAs, then it's going to be really small images for testing. And then those TMAs are coming from 10 to 15 different sites that, we don't have the whole sections for those, but obviously they can be used for testing as well. And okay, so the next question is, okay, what scanners are in the training set? So
yeah, that kind of is about the other question too. So just to have everyone aware of the mismatch between training and testing, so that they are creating their models in a way that can generalize to that mismatch. So they're not surprised when the validation study is different scanners,
right,
or TMAs when they hadn't seen a TMA yet.
Right. And then one question that, just now that you brought up these scanners, Brandon, so do we need to Obviously, what we have is the research scanner. So we have a Perio 82, we've had access to Philips, but as a sort of like a research use type of thing. So do you think this needs to be done in a, like at least for the final validation in a sort of like clinically approved scanner? They're the same, but you know, the labels are different.
Well, you haven't told, you described the challenge. You haven't described anything that would have any, I would have any input to that. You didn't talk about this actually being a regulated device in the end. Who would be the owner? Who would be the sponsor, as we call it at the FDA, to make the submission? Because if that's the kind of level that you want to be at, then things like what you will say about the algorithm would become relevant. And one of the things that you have to say is, what images does it work on? And when you say it works on these images and that images, obviously you need to have the data to demonstrate that.
Okay. Okay. Yeah, that's a good point. So we can specify that.
Well, maybe the one thing to keep in mind would be, if it's not the device, i.e. the AI algorithm, but if it's the data set that would undergo regulatory review as a tool to develop potential future devices, that would likely be different. Of course, if we can use clinical grade images, let's say on a cleared scanning device, that would probably be optimal. The question is, if the tool is, for example, to develop or assess performance of an otherwise developed device, right? So if there would be an AI algorithm that was tested or trained on, let's say, an approved device, and this data set is used for performance verification, that depends on the intended use of the tool, i.e. the data set being created. And I'm not sure whether that can be answered that easily. So I don't know, Brandon, what are the rules regarding tools? Do they have to be?
Well, I think my previous answer is the answer, because in the end, if it's going to come to the FDA, the label will say what images the algorithm can be used for. And we call that the intended acquisition device, right? And if you're talking about a tool and it's just about the truth data, the validation data, or if it's about the actual algorithm itself, you're going to have that element to it. Now, certainly a company can generate their own data on a specific scanner for the regulatory submission, and demonstrating that it works on these other data sets is evidence. It might not be the sum total of what is needed, but it certainly demonstrates performance. But when we're talking about an algorithm going to market, it will have part of its label say what image acquisition device it can be used for, and that would impact the value of the regulatory data as a tool. And I couldn't tell you whether the center would want to clear data that didn't have that regulatory element, the intended scanning device part of it, because certainly the value is less in that case.
The value for device sponsor, right?
The value of the data for device application, right?
Mm-hmm. But
this you never know.
Yeah, but this can go away. Let's say if we were to, let's say these take these slides and then actually scan them, let's say with a couple cleared scanners, then we are fine on that aspect. Is that correct, Brandon?
Sure, we have colleagues and collaborators that could scan slides for you as well. I mean, that shouldn't be the ultimate limitation of what you want to do. It's about what effort you want to do. And certainly there's some effort in moving slides around in order to get them scanned. And, you know, it has to touch other people and be in other organizations, at least for those moments. So that's part of the considerations.
Right. Yeah, yeah, you're right. I mean, it's not a huge bottleneck where we can just send it to.
Ellie, where are you? I don't know where you are.
In BC, Canada. Yeah.
Okay.
Yeah. And yeah, I think that's, it's just that those are the things that it's good to know so that we can actually, when we are putting together the data sets, we make sure that we actually account for those. And it doesn't seem like it's a huge effort anyway, so we can send them to a couple.
Yeah. My questions for the others too then are really coming from that, my role here. What would I be looking for in a device submission or a data set submission? You know, the whole characterizing truth is one of the pivotal things in all this work. When from my desk at least, developing an algorithm, it's all plug and place kind of stuff these days, it seems like. And getting high quality curated data is at the top of the list of things to look for. And so things like sites where the data comes from, the acquisition devices, some patient metadata, you know, demographics, gender, race, some of those things, all of that stuff is always good. I don't know what clinical covariates would make sense in this space, but I have a data set that I am marching forward in this way. And they wanted, you know, me to make sure that I had cases that represented all the different cancer stages. And a couple other, I could probably pull up my inclusion exclusion criteria at some point and share it with the rest of the community, and could do it for you sooner than that. But those are the kind of things, you know, really characterizing the data is one of the easiest things to do if you do it from the beginning, and certainly one of the first things we look at in a device submission. One of the things I look at.
Right. Yeah, that's fair enough. Yeah, I think for again, for these cases, we should have a lot of information, so it's not like, you know, we'll have to go back and pull out the path reports to get those. So a lot of those.
Well, that's the key, right? You do that once rather than going back, because going back is never fun because someone has already looked at some of the cases, right?
Yeah. But those are great points that we can actually incorporate, and hopefully not a huge amount of effort on our end. So it seems like for the time being, so which is good. And I think there was that last question from you as well. Okay, how many algorithms in clinical validation and what image viewing platform, how many in vitro pathologists? Yeah, so those are questions for the time being, you know, how many. So we'll have to do proper power analysis. Obviously, I mean, how many algorithms? I don't know, because it depends on how many pathologists we would need. And then if it is like 15, that's going to be like 3 different algorithms, 100 cases. That's going to be, you know, it depends on the amount of effort that people put.
Yeah.
We may go with it like a sort of like maybe top 3 consensus type of thing as well. I mean, those are the type of things that we can consider at that point, depending on how the results, you know, how they end up. So this part, we have a bit more, I think, freedom as we will, you know, we're going to get the results, evaluate the models, see, you know, how they do, if they're completely agreeing with each other. So maybe that won't have, for some cases, I think that will be the case. But for the subset, which are the challenging cases, it is going to be desegmented in models, then we can sort of come up with ways to resolve those. But the viewing platform, Yeah,
How are you gonna show those annotations to the end user and collect the data in that study?
yeah.
That
So if we
It's something to
were,
be thinking about.
yeah, absolutely. So we have our own viewer, but obviously that's not cleared. So that's for research use only that we do. But if we were to go like, to make this something that has regulatory value, I think that has to be done with an approved viewer as well.
Yeah, so those are things to be putting on the plate to discuss with your collaborators. And because for every algorithm, well, for one algorithm, you have the without the algorithm viewing and then with the algorithm. So that's two sessions right there. But then for every new algorithm, depending on whether you have the same readers or not, it might just be another study set of or another session with the algorithm. But if you start having multiple algorithms, at some point, using the same pathologist over and over again, then you're going to start having some concerns about them seeing the same images over and over. But there's some fun study designs that can split that up into more manageable sizes that I'd be happy to share with you. In fact, I'll try and look at that while we're still talking.
Yeah, that would be fantastic. Yeah, that's a great point to consider. So obviously,
Ali, let me say one more thing.
Yeah.
If you could use a viewer that supported standardized image, or even if you were to create standardized images, whether it's DICOM or some other standard that other viewers could use, there's an opportunity there that if you could prove that you're showing the same pixels independent of the viewer, that has a lot of value. Now, the algorithm, just like it being linked to the image acquisition device, also linked to the viewing device. So that's just something to keep in mind from a regulatory standpoint.
Right. Yeah, that's a great point. Yeah, I saw that. Yeah, page. Can't remember what viewer they used, but we'll look into that. And also, that will be a discussion point with collaborators and see how that can be done with the viewers. Yeah, great points. Any other ideas or input? Yeah, I see that you shared the link as well.
What's your rough time frame that you were thinking about?
The time frame, I think I would say maybe a couple of months. Like, let's say if we shoot for, let's say September, maybe I think that's reasonable. We have most of the data, but obviously, I mean, this is extra stuff that we will need to do, like scanning with different devices and that sort of stuff. We haven't done that yet, simply because we need to want to get feedback first, and then we will start those efforts. And with Kaggle, seems like they're, I mean, overall, so we've had good discussions with them. They're engaged. So they're asking for updates, but we're trying to delay. Say we're trying to design it in a way that has has more value for the community. But I would say probably September is a good timeline, depending on if we can actually do all the things that we want to essentially check boxes.
Yeah, I think as Brandon pointed out and what's in the decision summary from Page, it's this, I mean, pre-data compilation part, right? To really have a robust, I don't know, feature clinical key characteristics, kind of like the analytical plan that you outlined, accounting for those elements may be very helpful. Um, I know you're very experienced in that field. You probably have a clinical collaborator, right? Meaning ultimately the distinction of the various histotypes in some cases has direct treatment implications, but in others it does not. So of course, the plan is very ambitious to distinguish 5 subtypes, even possibly. I think I like the idea or the concept that you leave it up to the developer to say, parse out the outliers first before you classify, or classify all 6, right? At least I understood it like you leave this up to the developers to get both points sort of accounted for. But maybe one thing to consider is to have like the one tumor that has distinct treatment as a key endpoint for, is that decision affected? Because ultimately, as Brandon pointed out, people always look for one kind of endpoint. Is it, the classic 97 or 95% or whatever you want to call it? But for the key decision that's dependent on the histo classification, I think that may be helpful to just not to change the design for that, but to account for it, to have a very good sort of primary endpoint. And yeah.
I think building on that, going to the sampling you discussed, you talked about task number 2, adding in 25 outliers. I would think of it a little bit more prospectively, that who is getting sent for this diagnosis? Are you really only sending the actual cancers, or are you sending some normals? Because in your description of the cases, I didn't see any normals. And if this is kind of at the front end, how big is that net? Is there a population of actual normals that will be diagnosed with this algorithm at the same time as the cancers are getting diagnosed, right? Are they part of the clinical population that the device is intended to be used on? Or is it all verified cancers that it's going to be used on?
So there was actually, in one of them, actually I included, so it was like fallopian tube. No, it's going to be normal tissue and also benign as well. That will be part of it. Yeah, and so that again, if a case is suspected of having ovarian cancer, let's say biopsy is done or resection done, it will go through this. It's going to be either like benign normal tissue, or it could be one of the cancer outliers, like rare subtypes, or one of the 5 major histotypes. So that's roughly the plan.
Right. So when we talked about stratifying your population across those cancer subtypes, you should have normal as one of those cancer subtypes, or as one of those categories.
Right. Yeah.
That category is often overlooked when we're kind of going through this algorithm development party and challenge. But when it goes out in the field, patients without cancer are going to be encountering this device, or this device is going to be encountering images from normal patients without cancer.
Right. Yeah.
So that should be one of your metrics.
Yeah, and then yes, sensitivity and specificity can be actually done just on normal cases as well. I think that would be important.
Well, I would say you have sensitivity for the normals. We call it specificity, but you would create that 2x2 table where one of the rows is just the no cancer patients and the other is everybody else.
Yeah. Okay. Yeah, so I think you're right. I mean, it's probably good to actually have that 7th category, right? And just lumping everything as like outliers, say normal, rare cancers, the 5 histotypes. So that's essentially what we want to do. And then it's up to the developers how they develop it, obviously. And in what order or all at once, I think that won't matter too much for us.
Do you know, Ali, how the Panda challenge enabled the data entry or data submission, like how they organized that?
So that was done, yes, I mean, so there are two ways to do this, and then Mariam can comment as well, but two ways. So one is that you host, let's say you put the data somewhere, people download everything, and then they just submit the results, like their labels. And that's one way to do it. A lot of challenges has been like that. The Kaggle platform is actually makes it easier, meaning that they host the data, and then this is good for reproducibility because then they actually submit their Python notebooks. And then, because eventually you want to reproduce the results, right? You don't really want somebody to just submit some results and then you don't know what the details are, you know. And the good thing about Kaggle is that they submit that notebook, it runs, the Kaggle automatically runs it on, let's say, on your validation set, whatever sets you have, and it gives, obviously, the tuning set, the developers can see the results, but for the rest, we can see the results. That's how it is. That's why we have that limitation of like the data that they can host. They've been like pushing us to sort of like, you know, down sample as much as we can, or make this study smaller. Panda, they were working, yes, they had like 10,000 slides, but it was, these were needle biopsies, not much, like, you know, the files were small. Overall, I think it was around like 400 gigabytes. But with like, you know, just 550 cases that we have, all sections, we're talking about 1.2 terabytes. So imagine if we go to like 2,000 cases, then it becomes a challenge for them to host the data. That's why TMA is sort of like, it's a good compromise for the powerful part of the test set, because we get more cases, smaller tissue, this so that, you know, we can assess the generalizability of the models.
So the preferred way of, let's say, data entry in the latter version, you would submit your final model and then that would be executed, or do you submit your labels, meaning your results?
You submit your final model. As far as Mariam, is that correct? So I think that's the case.
Yes. And the same hardware facility is used for running those models.
It's all Docker based or something, container programming.
So it might be containerized, I'm not sure. But as far as I know, they submit a notebook. Presumably, yes, it should be Dockerized, either Docker or Singularity, something. Otherwise, it will be difficult to use, yeah, and we'll run them. Mariam, any comments? Did they use Docker or Singularity?
I think they are using Docker in
Okay.
Kaggle. Yeah.
Okay.
So then despite, let's say, a programmatic approach for submitting your model, they will have to, or you will have to, provide what the model output format looks like, right? So let's say case ID plus, and then comes a string or a table or whatever. So that part, I think, is determining how you can analyze the data. So that, let's say, if it's a probability for being one of the 5 tumors, so there's probably a probability for an outlier and being a probability for the top tumor or something. I think that part probably requires a little bit of consideration, right? Because some may actually have a nominal output, let's say an endometrioid ovarian cancer or a pancreatic cancer, right? You're not asking for delineating what other tumor it is, but there's some variability in that output, so it may be worth considering what that would look like to upfront define it, right?
Yeah, you're right. I mean, eventually we will have to define it, otherwise we can't really evaluate the models, right? And then most likely, I think we will go with sort of like a vectorized probabilities, like, you know, what is the probability of histotype X, Y, Z, and then we can, I think, process it that way. Or a vector could be like 0, is just one hot, meaning that just one cell. If they choose not to report the probability, it could be just, you know, one of those elements will be one, showing what histotype it is. Yeah.
Maybe good to narrow that down, because depending on whether that is, let's say, finalized, you can probably derive, you know, the statistical methods of analysis, and thereby, you know, what the power prediction and all the points that Brandon mentioned would look like. It's almost like, you know, I don't know, engineering it to match what you're looking for in terms of output. And of course, that depends on the intended use, what that challenge is about. But
Yeah,
yeah, my comments about the performance metric were based on a categorical output. And you can certainly get more information about device performance with a probability output, because then there's still freedom to implement a threshold. But that's where you have a little bit more choice. And I'm always of the sort of be as quantitative as possible. So I'm a fan of the probability outputs rather than categorical outputs. But you know, you have to be invested in how to take that output, either in an algorithmic way, what to do with it, or in a, you know, methodological way, what you're going to do with it. Either way, you have to have a plan.
Yeah, that's a great point to consider as well. You're right. Yeah, that will affect the number of cases if we were to go with the, you know, with the probabilities, and how that's going to affect the, I think the clinical validation will become important. But the other option is we get that information, but then we set threshold, it's thresholded at some point, and for clinical validation.
Ali, the other option, you know, I mean, I presume you work with some of the TCGA data on ovarian cancer, and, you know, some of the genomic data, you know, meaning secondary data points, just similar to, like, you know, ancestral data, stage, etc. That may be extremely valuable, especially, I mean, a data set like you describe it would be enormously valuable to the field. So if, for example, some people have, or some of the women have homologous recombination deficiency, and some of the people participating in creating the cases would submit those scores, you could even envision, you know, other uses of the data set that go beyond, let's say, those initial endpoints. But, you know, if those data sets would be available for, you know, later on use, you could use the same data set with an additional, let's call it iteration, with some molecular data or some TMB data, or, you know, more comprehensive molecular aspects that would make that, you know, like higher applicability for different questions, not just the histotype. But that's, of course, up to you. But I just thought that, you know, what would be probably the most clinical important question right now, it's, you know, of course, the histotype, and that you know that it's ovarian. But then, you know, once you have that point, you know, usually when we issue our path report, then the oncologist comes back and has additional questions, you know, which therapy line, what additional components to can be derived from that.
Yeah, that's a great point. Yes, I mean, we thought about that. I mean, so part of it, I mean, TCGA only includes high-grade serous, so it's not, so that's the problem. So it ignores the other 4 subtypes. So for that reason, and also TCGA, I mean, the ovarian set is not that great. So, I mean, there's slides. Asian slides. I think they were off of like frozen samples, I think, as far as I remember. Anyways, I have to double check, but there were issues with those. But yes, we thought about, genomics, obviously that's our focus as well, the link between, histopathology and genomics. But then data is in bits and pieces. We were thinking of actually including prediction of survival, which could become interesting because then we can look at HRD and also, signatures potentially in which patients could benefit from it. But the limitation is that from the competition perspective, Kaggle is, we had those three. They said that we need to drop one. So we decided to, meaning that the survival analysis, but that is sort of like the outcome is not guaranteed that anybody would come up with something, an algorithm that would stratify the patients into different groups. So again, logistics that also, limits the scope a bit on those aspects. But I think later on, so I think studies for some of these cases, what we can make sure is that we include some cases that we have some sort of genomics data or whatever molecular data that we have. And then eventually won't use it as a competition, but we can release it at some point for future studies. I think that's a possibility that we can assess.
Is that kind of data expected in the patient record?
No, because these are not in practice, we're not using them for diagnosis, or these are more research use, you sequence the cases or you see, what drugs could benefit, patients can benefit from them. But, from the guidelines perspective, there's not much to do, but it will be interesting as an example to identify BRCA mutant cases, because that has implications clinically. We will see if we can actually pull those out as well. So for a subset of cases, we may have that information. But that's a great point. So Hossein, can you make a note of this as well? But I think within our data set, we can make sure that at least we include some of those, even though we can't have another set of tasks for the challenge. But that could be a second one, at least with the same data, we just released just the, let's say genomic data or some more clinical data.
Yeah, my question about whether it was done clinically was because if it was, then someone here at the agency might say, well, make sure that you have some positive for that biomarker and some negative for that biomarker to demonstrate your algorithm is robust to that. And they wouldn't actually make you do a subgroup analysis or anything on that, but that at least I don't think so, but it's really about making sure your study population represents and covers all these different covariates of your clinical population.
Right. Yeah.
And that's another reason why kappa is not the best here, because all of that stuff wreaks havoc on the kappa actual numbers. You can compare two algorithms using the exact same data with kappa, but when you start trying to generalize across studies, that's where trouble starts.
Yeah, that's a great point. But what I can double check is that whether, from the clinical perspective, there is anything else that needs to be considered. I think that's a great point. And double check to make sure that we are not excluding anything just in case.
Brendan, we didn't have time to go over the split plot designs, but it may be worth, as, maybe a potential follow-up. Maybe once everyone on the call had time to read this. I know we discussed it before, but that may be actually a very smart combination to decrease the total number of cases and invest the time rather to count for different scanners or other, higher efforts in terms of clinical data quality, by accounting for that. And it may be great to, I don't know if you or Stephen or Nancy or some of the other people on the papers would be willing to share at least some of that with this group. But
I'm giving that talk to your group. I've given that talk internally. They're at my colleagues across the way are asking me to have a video for it so that they can share that with industry. But
we could just make this a session and provide the video,
that ICC is a good outlet for that.
I think
Maybe
it's
summer.
it's highly applicable to a problem as broad as this. I mean,
Yeah, there's another study that I didn't put there because I didn't really love how I was included near the end in compiling the paper, but that's neither here nor there. But we did a split plot study as exampled in those two papers I put out there. And, they wanted to test all these different modalities, which is that same kind of idea as testing multiple algorithms, because you kind of need a full study for each one of those random effects, if you will, or even fixed effects. And, I think it was just a little bit ahead of my time. All my methodology wasn't as ready for prime time, maybe, and they certainly were a little hesitant to follow me down that path or wait for me. And so here's the paper. It was in a mitotic. And I think the biggest challenge was that it was in a mitotic figure counting study, and that data was just very, I don't know if you've done mitotic figure counting with multiple readers, but the more readers you have, the more candidate mitotic figures you get. And so things just didn't look like they were at the performance levels that they wanted to publish. And but I mean, I think we did some good work in that paper with the split plot study design to allow us to interrogate many different imaging or viewing modes. They wanted 20x, they wanted 40x, they wanted this stain and that stain, and you start building that into a multifactorial study. There's so many things to study that time is, you can't have everybody read everything. That's the bottom line. And the split plot study goes a long way into divvying up the workload.
Yeah, absolutely. So I think that
Joe, you always keep a good eye on the clock.
no, I'm not.
Oh.
I can ignore the clock. If it is as exciting as this, there's no time.
I have to go though.
Okay.
Everybody.
All right.
Again, thank you. Really
Good
appreciate
luck,
it.
Ali. Good luck, Ali.
Thank you.
Let's talk soon. Bye.
Thank you.